Hello, this is Mrs. Kohler. In this unit, we are going to start talking about DNA. In the last unit, we talked about cell division and how the DNA divides. In this one, we're going to talk about actually what it looks like, how your body makes more of it, and how it turns the DNA into you. So before we actually talk about DNA itself, though, I want to talk a little bit about the history of DNA, because it's important to understand that science is a process. We don't just magically know everything instantaneously. It takes time to learn new things, so science can change over time. So we're going to talk about how our idea of DNA changed as you go through different years. So the first scientist we're going to talk about in this PowerPoint, now you're going to do a timeline that includes some others. But the first major one we're going to talk about, his name was Frederick Griffith, who was doing his research in 1928. And what he did, now he took mice and he knew that there was a particular bacteria that had two different strains. One strain was harmless. In other words, the mice could get it inside of them and it wouldn't hurt them. And those were alive. And then he took another strain of the same bacteria that was dead, but if it would get into mice when it was alive, was harmful. It would make them sick or it would make them die. So he injected mice with a combination of these. So he took live bacteria that didn't make them sick, mixed them with dead bacteria that would make them sick, and put them in the mouse together. And what happened was, even though the harmful bacteria were dead, the mice died anyway. So Griffiths decided there must be something inside that dead bacteria that the live bacteria got into themselves to make them harmful. And he called this thing a transforming factor. Transforming because it transformed them from harmless to harmful. But he didn't know what that transforming factor was. He just knew that it existed. So that was in 1928. In 1944, Avery ran a test to see what Griffith's transforming factor was. So at this point, scientists were pretty sure that it was either protein or DNA, but they weren't sure which one it was. So he tested the DNA, and he tested proteins, and he did this using a type of bacteria. And his conclusion was that DNA was the transforming factor. So DNA was the thing that was able to pass characteristics on from one organism to another. Now, a few years later, Hershey and Chase decided to do some more research in order to see if what Avery found was actually true. Remember, for science, you want to get the same results repeatedly in order to confirm that the first result wasn't just a fluke. So Hershey and Chase were repeating this experiment, but they used something a little bit different. They used a bacteriophage. Bacterio would be bacteria, or a type of virus that infects bacteria, to confirm what Avery did. And they confirmed that DNA is what transmits genetic information. And of course, in the 1950s, Watson and Crick discovered the actual structure of DNA, which we'll talk about later. But what I want to do with you right now is actually walk you through one of the experiments that was done. And I'm going to walk you through the Hershey-Chase experiment. So I said that I was going to show you the Hershey and Chase experiment that showed that DNA is the transforming factor in organisms. So here you have a bacteriophage. And remember, a bacteriophage is a virus that infects bacteria. Now, the nice thing about a bacteriophage is it's only made of two things. The first is protein, and the second is DNA. So what they wanted to find out was, what is it in the bacteriophage that transmits genetic information? Is it the protein or is it the DNA? So when you're using something that only has those two, then you know that it can't be anything else. It can only be one of those two. So here's your virus, kind of looks like a little robot. And we're going to click on it to begin. So here it is attaching itself, and then it injects its genetic material, it multiplies, and then it bursts out of the cell again. So their question was, which part of it was it that caused the cell to start making new viruses? So they made 2 batches of bacteria. The first batch, which is this one right here, they treated the protein with a radioactive isotope. And in the second batch, they treated the DNA with a radioactive isotope. So the idea is, see which one of these gets injected into the bacteria, and then we'll know which one is the genetic material. This is what batch 1 looked like. You can see that they're glowing around the outside. That's the protein coat that's coated with radiation. And here you can see that they're glowing inside the heads. That's where the DNA is, so that's the part that has the radiation. Now, what they did was they took these, they stuck them in a blender, they mixed it up, and then they put it in a test tube. Now, once they had them both in a test tube, they need a way to separate the protein from the DNA. So they take them, they put them in a centrifuge, and the centrifuge spins it around so that it separates. So you have liquid there and you have bacteria there. Now they test the radiation level. So go up here. For batch 1, had high radiation in the liquid, but low radiation in the bacteria. So they know that in batch 1, the radiation did not get injected into the bacteria. So that was the one with the protein coat. So that could not have been the genetic material because it didn't make it into the bacteria. For the second one, you have low radiation in the liquid, but high radiation where the bacteria are. That means that the part of it that had the radiation got injected in, and in that case, it was DNA. Up here, this had the high radiation, and down here, this had the high radiation. So as you can see, based on this experiment, it's clear that it wasn't the protein that was the genetic material, it was the DNA that was the genetic material. And that's how that was discovered.